Hello, welcome to Biology, the scientific study of life. Again, protein conformation and function. Once we have learned the amino acid sequence of polypeptide, what can it tell us about protein conformation and function? The term polypeptide is not quite synonymous with the term protein. Even for a protein consisting of a single polypeptide, the relationship is somewhat analogous to that between a long strand of yarn and a sweater of particular size and shape that one can knit from the yarn. A functional protein is not just a polypeptide chain, but one or more polypeptides precisely twisted, folded, and coiled into a molecule of unique shape. Notice this picture. Notice. It is the amino acid sequence of a polypeptide that determines what three-dimensional conformation the protein will take. When a cell synthesizes a polypeptide, the chain generally folds spontaneously, assuming the function conformation for that protein. This folding is driven and reinforced by the formation of a variety of bonds between parts of the chain. Which in turn depends on the sequence of amino acids. Many proteins are globular, roughly spherical, while others are fibrous in shape. Even within these broad categories, countless variations are possible. A protein's specific conformation determines how it works in almost every case. The function of a protein depends on its ability to recognize and bind to some other molecule. For instance, an antibody. A protein binds to a particular foreign substance that has invaded the body, and an enzyme, another type of protein, recognizes and binds to its substrate. The substrate the enzyme works on. This is a space-filling model. Notice that shows more clearly the globular shape seen in many proteins, as well as the specific conformation unique to lysozyme. Okay, we have learned in chapter 2 that natural signal molecules called endorphins bind to specific receptor proteins on the surface of brain cells in humans, producing euphoria and relieving pain. Morphine, heroin, and other opiate drugs are able to mimic endorphins because they all share a similar shape with endorphins and can thus fit into and bind to endorphin receptors in the brain. This fit is very specific, something like a lock and key. Thus, the function of a protein, for instance, the ability of a receptor protein to identify and associate with a particular pain-relieving signal molecule, is an emergent property resulting from exquisite molecular order. 4 levels of a protein structure. In the complex architecture of a protein, we can recognize 3 superimposed levels of structure, known as primary, secondary, and tertiary structure. A 4th level is a quaternary structure, arises when a protein consists of 2 or more polypeptide chains. Okay, this is the following 2 pairs describes these 4 levels of a protein structure. Notice levels of a protein structure. First one is primary structure. The primary structure of a protein, it is its unique sequence of amino acids As an example, let's consider transthyretin, a globular protein found in the blood that transports vitamin A and a particular thyroid hormone through the body. Each of the four identical polypeptide chains that together make up transthyretin is composed of 127 amino acids. Shown here is one of these chains unraveled for a closer look at its primary structure, a specific one of the 20 amino acids indicated here by its 3-letter abbreviation. For instance, GLOW, short term, and LSP3 LSR, this one short term of this skeleton, to short term of glycine, phosphate, alanine, valine, methionine, phenolene. This skeleton non-polar, polar, serine, threonine, cysteine, tyrosine, asparagine, glutamine. The first letter of the 2 or 3 letter is the symbol of amino acid. So this or these are connected together to contain amino acids to 127. All these, the skeleton, closer look of these skeletons. Okay. Abbreviation, 3-letter abbreviation occupies each of the letter of the 127 positions along the chain. Notice that, so long. The primary structure is like the order of letters in a very long word. If left to chance, there would be 20 different ways of making a polypeptide chain 127 amino acids long. However, the precise primary structure of a protein is determined not by the random linking of amino acids, but by inherited genetic information. Notice the end is a carboxyl end. Notice one. Secondary structure, notice the secondary structure here, amino acid. The end is amino acid subunits. Most proteins have a segment of their polypeptide chains repeatedly coiled or folded in patterns that contribute to the protein's overall conformation. These coils and folds, collectively referred to as secondary structure, are the result of hydrogen bonds between the repeating constituents of the polypeptide backbone, not the amino acid side chains. Both the oxygen and the nitrogen atoms of the backbone are electronegative with a partial negative charge. Notice this one. A pleated sheet and a helix. Notice a pleated helix. Okay. Where is the... notice that the weakly positive hydrogen atom attached to the nitrogen atom has an affinity for the oxygen atom of a nearby peptide bond. Individually, these hydrogen bonds are weak, but because they are repeated many times over a relatively long region of the polypeptide chain, they can support a particular shape for that part of the protein. One such secondary structure is the helix. Which means alpha helix, a delicate coil held together by hydrogen bonding between every fourth amino acid shown above here. For transthyretin, although transthyretin has only one alpha helix region, which means other globular proteins have multiple strategy of alpha helix separated by non-helical regions. Some fibers, proteins such as keratin, the structural protein of hair, have alpha helix formation of other over most of their length. The other main type of secondary structure is the B-pleated sheet as shown above. In this structure, two or more regions of the polypeptide chains lying side by side are connected by hydrogen bonds between parts of two parallel polypeptide backbones. Pleated sheets make up the core of many globular proteins, as is the case of transthyretin, and dominate some fibers proteins, including the silk protein of a spider's web, here as it is shown here. A spider's web, the teamwork of so many hydrogen bonds makes each spider silk fiber stronger than a steel strand of the same weight. Third one is a tertiary structure. Superimposed on the patterns of secondary structure is a protein's tertiary structure shown above for the transthyretin polypeptide, rather than involving interaction between backbone constituents. Tertiary structure is the overall shape of a polypeptide resulting from interactions between the side chains or groups of the various amino acids. One type of interaction that contributes to tertiary structure is somewhat misleadingly called hydrophobic interaction, as a polypeptide folds into its functional conformation amino acid with a hydrophobic nonpolar side. Chains usually end up the closures at the core of the protein out of contact. The core of the protein, a protein out of contact with water. Thus, what we call a hydrophobic interaction is actually caused by the action of water molecules, which exclude nonpolar substances as they form hydrogen bonds with each other and with hydrophilic parts of the protein. Once nonpolar amino acid side chains are close together, van der Waals interactions help hold them together. Meanwhile, hydrogen bonds between polar side chains and ionic bonds between positively and negatively charged side chains, also their cumulative effect helps give the proteins a unique shape. Notice that SS. The conformation of a protein may be reinforced further a covalent bonds called disulfide bridges. Disulfide bridges from where two cysteine monomers amino acids with sulfhydryl groups SH, notice this SS, on other side chains are brought close together by the folding of the protein. The sulfur of one cysteine bonds to the sulfur of the second, and the disulfide bridges SS, notice that, rivets parts of the protein together. Notice the yellow one represents SS. These lines, all of these different kinds of bonds can occur in one protein as shown above in a small part of a hydrophytical protein. Third one, catenary structure. Some proteins consist of two or more polypeptide chains aggregated into one functional macromolecule. Catenary structure is the overall protein structure that resulting from the aggregation of these polypeptide subunits. For example, shown above is the complete globular transthyretin protein made up of its four polypeptide, 1, 2, blah, blah, which is a fibrous protein that has helical subunits. This one. Subunits intertwined into a larger triple helix. Notice this one inside this, and this third catenary structure. Grease string dissociates collagen fibers to their function as the example collagen shown in the right, which is fibrous protein that has helical subunits intertwined into a larger triple helix, giving the long fibers grease strength. This suits collagen fibers to their function as the guidas of connective tissue in skin, bone, tendons, ligaments, and other body parts. Collagen accounts for 40% of the protein in a human body. Hemoglobin also, notice collagen, this represent collagen, and this chains beta. Hemoglobin, okay. Notice also hemoglobin, the oxygen binding protein of red blood cells shown below, is another example of a globular protein with catenary structure. It consists of 4 polypeptide subunits, 2 of one kind, A chains, and 2 of another beta or beta chains, both A and B. Notice A helix and B pleated sheet. Both also unit consists primary of a helical secondary structure. Each subunit has a non-polypeptide component called heme with an iron atom that binds oxygen. Thanks for listening.